Hello friends, this is Neha Antal, Assistant Professor from IMS Ghaziabad University Courses Campus. Today I am here to discuss a topic on bacterial staining. Now we will study bacterial staining. What is bacterial staining or further? First we discover what is bacteria. As we all know, bacteria are microscopic organisms. They are colorless for the most part. In order to visualize them to study their structure, shape, and other structural characteristics, it becomes necessary to make them more easily visible. To make them visible, we use staining or any colored object. What is a stain? Stain is a dye used to color the living or a dead organism, or we can say it is an organic compound which is either positive charge or negative charge or carrying both the charges. Stains. Based on the charge, we can specify stains in three parts: basic stain, acidic stain, and neutral stain. Basic stain or dyes are with positive charge, acidic stain dyes with negative charge, and neutral stain dyes, as it name suggests, stains with both charges. Function of stain. What is the function of stain? As we have studied, or we will study, the stains are of three. Stains are of categorized into different parts, like for example, simple staining, differential staining, and special staining. Simple staining, as its name suggests, only where only one dye is used, and differentiation among bacteria is impossible because only one dye is used here. Example: simple staining. It gives same color to all bacterial cells but provides color contrast so we can differentiate bacterial cell and other cells. Next one comes differential staining. Differential, as its name suggests, differential, making a difference. Where so in this staining more than one dye is used and differentiation among bacteria is possible. And differentiation among bacteria is possible because it is imparting different colors to different bacteria. For example, gram staining, acid fast staining. Next one comes special staining, where more than one dye is used and it is used to study special structures like bacterial capsule, bacterial spore, or a flagellus staining. Unspecial or negative staining, here the cells remain clear and background is colored to create a contrast to aid in the better visualization of the image. Example here, we use Indian ink and necrosine and special staining. Principal staining, why we stain the bacteria, as we have discussed, it is giving differentiation or it is providing visibility to the bacteria. Staining, every staining methods have its own common principles, like for example here we are you are saying basic stain, that is positive charge. Here it is a positive charge, to stain negative charge, because that stain is carrying positive charge, so it will make a complex with a negatively charged molecule. So here it is used to stain negative charged molecules of bacteria, mostly used because most of our cell surface is a negative charge. Next comes to acidic stain, which is negative charge. So if we have to stain positive charge, we will use acidic stain. They stain positive charge molecules of bacteria. uh, in acidic stain, we study bacterial capsules. As I have discussed, cell surface is negatively charged, so mostly basic dyes are mostly used. What are the basic requirements for staining? First, you need clean glass slides, Then you need bacterial culture, which is 24 hours of growth, and inoculating loop to make a suspension or to transfer bacteria culture into onto slides, then Bunsen burner for heat fixing, and or preparing a smear to heat dry a smear, pencil marker to mark the bacterial smear. Okay, and basic initial step before staining. Before staining, we have to prepare a thin opaque layer of a bacterial culture that is known as smear preparation. So in this method, we firstly make a suspension of bacteria culture, and which means if you have a solid bacteria culture, then you will mix some amount of water and make it a liquid material, liquid culture. Then one drop of bacterial suspension is put on the slide, and then you have to move it in a circular fashion, then air dried and then heat fixed, and the resultant preparation which we obtain is known as bacterial smear, which appears dull white. As you can see here it is taking a suspension of bacterial culture with the help of a inoculum, or we can inoculation loop. Next, we are putting a small drop of water and making in a circular way. Then here it is look like this. When this circular area or a culture heat dried or get dried, it will look like this. This structure looks like a opaque opaque culture.